All right, good afternoon. I want to welcome everyone and thank you for joining us for today's Adventures in Field Research, the Spring 2021 seminar series. We're happy today to be joined by Dr. Andrew Steen from the University of Tennessee in Knoxville. And Dr. Steen is going to give us a presentation he calls What to Eat When You're 100,000 Years Late for Dinner, the Secrets of Slow, Uncultured Microbes in Deep and Cold Places. So we are looking forward to hearing what Dr. Steen has to say, and then everyone can follow up with some good discussion and questions at the end. So with that, I'll turn it over to Dr. Steen.
All right. Well, thank you very much for inviting me here. I'm a little embarrassed to have sent that, like the one picture of me in a nice shirt with a background, because usually I like to send pictures of me actually having fun in the field, which is where I like to be. So I am I'm a professor in the Department of Microbiology and Earth and Planetary Sciences at University of Tennessee. And the reason that I am associated with both of those departments is that my work is partly about microbes and partly about their role on affecting the geochemical cycles of the Earth. And I'm particularly interested in microbes that live a little bit far away from where we are used to seeing them. So there are microbes all around us all of the time. They are on our skin. They are in the soils. They are, anywhere you can look. If you had a camera that could only take pictures of microorganisms, that could only see microbes and nothing else, it would take fairly normal pictures of the world, because everything in the world is covered in microbes, and that's fine. Living in the soil or living on a plant leaf or something like that is not very much like living in the human body. So the vast majority of these microbes don't actually hurt us in any way. They're helpful in as much as they help keep the Earth environment the way we like it. So I'm going to talk about some microbes that are living at the limits of life. And sometimes when we think about the limits of life on Earth, we think about like hot environments or something like that. And I'm particularly interested in microbes that live in the cold, and particularly in microbes that live slow lifestyles. So how slow of a life can you live? And then how do those microbes affect Earth's geochemical cycles? And to explain that, I'm going to talk a little bit about my work in the ocean and in estuaries, and then my work in the Arctic. So I want to start off with a place called the White Oak River Estuary, which is not too far away from where we are here, or at least where I am. I don't know where you all are out in the internet. So the White Oak River Estuary is this little estuary about maybe about 10 miles long, and it's fed by this smaller river. It's in coastal North Carolina, and it's, looking at the map, it's not a place that maybe stands out on the map for you, but it's quite lovely. I really enjoy going down there, and the reason I like going down there is because the mud there is really interesting. So about once a year, my lab and I, and usually some of my collaborators, go down to the White Oak River Estuary, and we collect sediment cores. And the reason we do that is that we want to know who lives in this mud, and more accurately, what kinds of microorganisms live in this mud. And then what are those microbes doing? So, how can we find out about that? Well, it turns out that microbes breathe just like we do, and those of you all that are taking biology will be certainly familiar with this. But we breathe in oxygen and we breathe out carbon dioxide. This is a scanning electron microgram of some microbes that are attached to sediments. The little spherical things are microbes here, and they're maybe 1 micrometer in diameter. And they also breathe in oxygen and breathe out carbon dioxide. But if they're microbes that live in these muds that I showed you, a couple minutes ago, the only source of oxygen is from the surface water, which has oxygen in it, and then ultimately the atmosphere. And so, to find out, let me change my order here a little bit. Because the only way that oxygen can get into these sediments is from the surface water, then if we look at an oxygen profile, and I've cut off the vertical axis labels here, but this is millimeters down into marine sediment. So this hash mark is, I'm going to say 5 millimeters deep in sediments. Yay much. If we measure oxygen concentrations in the sediment, we see that they go down. In fact, they go down to 0. And that tells us something is consuming that oxygen, and it must be microbial life. So to make measurements about that, we go out into the white oak, we collect these sediment cores. One reason I like working out in the white oak is that it's very easy to collect sediment cores. So we go out, hopefully on a nice day, and just hop in the water and take these cores, and we swim down about 4 feet or so, and we just press them down into the water with our hands. It's about the easiest sampling there is. The mud is so sloppy and gooey that we can just press the cores down with our hand, and then we put a stopper on top of them and pull them up, and that keeps the mud from falling out. And then we load them up onto this little boat, and we do that all day and end up with a whole bunch of nice mud that we can mess with. So once we've got the mud, we take it into a lab. I've been working with people at the University of North Carolina, and they have a coastal lab, so they're very kind and let us use their lab. And so we can do all kinds of chemical measurements on these sediments to measure things like how fast oxygen is being consumed, and therefore how active the microbes in these sediments are, how fast they're breathing, essentially. The thing about microbes, and this is one of the things that I love about microbiology, is that humans and whales and monkeys and dogs and almost every other kind of eukaryotic organism, so organisms that have cells that have a nucleus in them, can breathe oxygen and breathe out carbon dioxide. And that's about as far as eukaryotes go for the most part. Microbes can do all kinds of breathing, and so they can breathe in oxygen, they can breathe in nitrate, they can breathe in sulfate, they can breathe in different kinds of metals. And when I say breathe, what I'm talking about actually is the process of moving electrons between molecules, or between atoms properly, to gain energy. And so we can only breathe in oxygen, but microbes, we can breathe in oxygen, use that as a place to put extra electrons. Microbes can do that with nitrate and sulfate and all kinds of other compounds. And so we look at the concentrations of a bunch of different chemicals in these sediments to look at the different kinds of microbial metabolisms that are happening in sediments. So this is a plot from the White Oak River of sulfate concentrations, which are in the unfilled circles, and then methane concentrations, which are in the black dots. Excuse me. And we see that over about 40 centimeters, so about a little more than a foot and a third or so, microbes they have breathed all of the oxygen. They've taken all the oxygen up within a couple of millimeters. And then over 40 centimeters, they are taking up all of the sulfate and converting it into sulfide. And then once they have done that, they can do a different kind of metabolism that involves taking in carbon dioxide and breathing out methane. And so we really care about this process because, well, for a bunch of reasons, but one reason is that both carbon dioxide and methane are, of course, really important to the greenhouse effect. They're both greenhouse gases. So, in fact, one of the big controls on, and I should say this is just a diagram of parts of Earth's carbon cycle here, but what you can see is that plants do photosynthesis and take organic matter, or sorry, take carbon dioxide out of the atmosphere, and they turn that carbon into plant biomass. And, every carbon in a tree used to be carbon dioxide, and a lot of that carbon winds up in the soil. And once it's in the soil, so that's, then it's out of the atmosphere. But once it's in the soil, then that soil carbon can be eaten by microbes and turned into carbon dioxide. And then deeper down in soils and marine sediments, there's methane producing processes that release methane, or potentially release methane, into the atmosphere. So if we want to understand the carbon cycle of the entire planet, then we need to understand what's going on with microbes in, at the smallest scales in places like marine sediments. And this is just to highlight the point. These are records of carbon dioxide and methane, carbon dioxide from the year about 1700, I think, and methane from year 0 CE. And what you can see is both of them were sort of kicking along relatively statically until 1850, 1900 or so. And then both of these things take off. And that's, of course, due to human activities. Carbon dioxide is mostly about burning fossil fuels. Methane is partly about fossil fuels and partly about changes in land use. But in both cases, we're putting a lot of carbon dioxide and methane in the atmosphere. That's increasing the greenhouse effect, and that's causing climate change, right? So we would like to know about the microbes that are involved in those cycles. But I also said, I'm interested in this sort of just purely for the microbiology also. Because it turns out that these cells must live incredibly slowly. So I am showing you again one of these sediment cores we've taken from the White Oak River estuary, and it's maybe about 1 meter long or so, so like maybe 3 feet. And if you look at the top of this sediment core, then that sediment was deposited probably like this year. Down at the bottom, those sediments might be about 400 years old. And the microbes that are in this, as I showed you a couple plots back, those microbes are producing methane. Those microbes, the way they get into that sediment is they get deposited at the surface of the sediment. And then over the course of 400 years, they get buried as new sediment lands on top of the bottom of the estuary. And so the microbes that are eating organic carbon have not seen any fresh food for like 400 years. And that means there's not a lot of fresh food there. And that means that they must be eating very slowly and therefore living very slowly. So if we want to understand carbon fluxes out of marine sediment, so how carbon moves between the oceans and the atmosphere and marine sediments, we need to know about what these microbes are eating. So, I gave you an incomplete chemical reaction, and I'm going to give you a slightly more complete chemical reaction here, which is that we make carbon dioxide by combining, let's say, a hamburger with oxygen to produce carbon dioxide plus water, and then that releases energy, and we use that energy to run around and jump up and down and do whatever we do with our days. So this is the question, is what kinds of organic carbon are these microbes using? But we have a problem with this. So if you are what we might call a classical microbiologist, you say, we want to know what this microbe is doing. Well, this is very easy. We have been doing this for hundreds of years. What we are going to do is grow the microbe in pure culture. That means that it's the only thing growing. It's the only organism growing, in this case on a petri dish. and we're going to let the microbe grow, and then we can poke at it, right? We see if it likes to eat. We don't actually test hot dogs and hamburgers and carrots, but we test things like, we might add specific proteins, we might add sugars, we might add fats, we might see what the growth of the organism looks like at different temperatures or at different pH or whatever else conditions we are interested in, right? And that works great. We know a whole ton about how microbes work from doing these kinds of experiments. What I think of, and I should say my bias is I come out of chemistry and earth science, and I'm sort of learning the microbiology a little bit as I go. So I think of it as just, we get a microbe kind of as a pet and we poke at it to see what it does, right? And the problem is that the vast majority of microbes that are out in the environment don't want to grow in pure culture. So this is just a plot of how similar microbes, it's a histogram of how similar microbes in the soil and in the ocean and in a whole bunch of different environments combined together, how similar those organisms are to microbes that have been grown in culture. And so basically we say that if they are 97% similar in terms of the sequence of a specific gene called 16S ribosomal RNA gene. If they're more than 97% similar, then we say they're the same species. So here we see that in these soils, only 10% of the microbes in the soils were 97% similar or better to an organism that was growing in pure culture. 90% were a different species. Again, in the ocean, we got 13%. In this mix of different environments, we got 4%. And this is not for lack of trying, or at least it's mostly not for lack of trying. There are people, and I am not one of them at all, but there are people who are experts at growing previously uncultured microbes. And it's insanely hard. You have to be extraordinarily careful. I can't grow a plant in a pot, so I don't really want to mess around with that line of research, but I have tremendous respect for the people who are looking at these organisms that are dissimilar from what has been grown in culture and trying to bring them into culture. But it's slow work, and there's a lot of microbes out there. And so just culturing is not going to cut it. So what do we do instead? We use the DNA of microbes in the environment to figure out what they're doing. And so this is sort of a general plot of the process of creating what's called a metagenome, which is to say, if a genome is all of the DNA that's in one organism, a metagenome would be all of the DNA that's in an entire environment. So what we do is we go to our environment. Maybe it's the human gut, maybe it's the ocean, maybe it's soils, maybe it's whatever else. And we extract the DNA chemically. We usually split open the cells and we extract the DNA chemically. And we do a little bit of processing stuff to it. So we've got pure DNA, we chop it up into little pieces, and we put it into a DNA sequencing machine, and this DNA sequencing machine does some chemistry that is so astonishing to me that I just almost can't even believe that it works. But it does, and these, for the low price of $10,000 or so, we might get, depending on what system you're using, like 10 gigabases. So basically 10 gigabytes of DNA data, which is just a stunningly large amount of DNA data. And then, because that's a whole lot of data, we have to do all kinds of crazy computational steps that I'm not going to describe, but we have to take these little short strands of DNA, and then we have to sort of pile them on top of each other to see how they go together. This is a little bit like if we went into a library and we chopped all of the books into individual pages and mix them all up and then try to piece them back together to figure out which pages go next to each other and ultimately try to reconstruct all of the books in the library, right? So that takes a ton of computational power, and I've been lucky to work with some people that are supercomputing experts, and we've done some stuff that's like computationally amazing. I shouldn't say lots of people in the community do stuff that's computationally amazing. Incidentally, at Oak Ridge National Lab up in Oak Ridge, they have what used to be the fastest computer in the world. Now it's the second fastest computer in the world. And a big chunk of what that computer does is this kind of work, working with DNA data sets. So once we have done that, we can figure out what all of the genes, or we try to figure out what all of the genes in an organism are. And then we see how we know what chemical processes those genes code for. And we try to put all of those processes in together to make sense of the metabolism of the organism. So this is just an example of the metabolism of one organism that a colleague of mine, Jordan Bird, tried to put together and I'm going to talk about this one a little later because it's pretty interesting. So that's sort of the basic approach, is we're trying to, because we can't really directly test what microbes in the environment are doing, we're trying to use DNA to sort of reconstruct what they might be doing in the environment. So we've done a lot of that in the White Oak River, and I'll tell you in a little bit about what we found. But what other places, one of the things we found is that there are these microbes that they're doing stuff that's in sediments that haven't seen the light in 400 years. I love this picture. I snapped this from Google Earth because it emphasizes that the Earth really is a water planet. So really, we can find if you are in the International Space Station, depending on your orbit and where you are in it, you can find views where you're basically only looking at ocean, right? We can see Baja California up here, and New Zealand down here, and Hawaii would be somewhere out here. So there's a lot of ocean on planet Earth. And we would like to know what is living in the sediments underneath these oceans. So to do this, we can use what are called drilling ships. And there's two major scientific drilling ships in the world. There's a whole bunch of drilling ships that are owned by oil companies that they use to explore for oil and dig oil wells. There's two big scientific ones. One's called the Joides Resolution and is owned by the United States. One is called the Chikyu and is owned by Japan. And so what these do is they sail out to wherever you want to sail them in the ocean, and they can drop their drilling rig down to the bottom of the ocean, which might be something like 4 or 5 miles. And then they can drill another more than a mile deep into those sediments beneath the bottom of the ocean. Again, this is one of these sort of technical feats that I kind of can't believe actually works, but it does. And I should say, I'm going to show you a bunch of cool pictures from life at sea in a little bit that I've taken, but I don't go on these ships, and it's a little weird because that's how you collect these. I am interested in microbes in the deep subsurface, and that's how you collect these deep subsurface samples. But these cruises are 3 months long, and I have a family, and I do not want to leave my family for 3 months. So I often go on these field trips to, North Carolina, or I'm going to show you a bunch of pictures from the Arctic. I've been to the Amazon. I've been to a bunch of cool places, but I won't go for more than 2, 3, maybe 4 weeks at a time. And I mention that just because I know that you've had this is a series about field work, and there are all kinds of neat things you can do with field work, and I love doing field work, but I also think that it's really important to figure out for yourself why you are doing field work and how that fits in with the bigger picture in your life. So when my kids are teenagers and they're sick of seeing me around, this one is already, I think, a little bit sick of seeing me around, then you know, I will probably try and get one of these drilling ships at some point. But for now, you know, that's not what I'm doing. So I depend on the kindness of my colleagues to collect these samples, and I work on them, and then I work on samples that I have collected from other places. But what's going on in the deep subsurface is really amazing. So this is a plot of cell concentrations, and annoyingly, these are in log, well, not annoyingly, these are in log units because these span such enormous ranges. So these cell concentrations go up to from, you know, 1,000 cells per cubic centimeter up to, it was 10 to the 9 is 1 billion cells per cubic centimeter. And then this is in log depth units. So 0 means 1 meter, 1 means 10 meters, 2 is 100. So we go down to not quite 1,000 meters depth. And what you can see is that even at 1,000 meters depth, there are, in some cases, lots of viable, lots of identifiable cells. So even in 1,000 meters depth, there are cells that are still living. And then I want to highlight this spot right here. So this is a plot of chlorophyll in the ocean surface. And so that's a pretty good indicator of how much, you know, how much photosynthesis is going on. So how much primary production is happening, how much food there is for the whole community to eat. And take a look at the South Pacific Gyre. There's very close to no chlorophyll in the water. And the reason is there's not enough nutrients to support really any kind of phytoplankton. Specifically, there's not enough iron. But what that means, there's no chlorophyll, there's nothing growing in the surface water. So there's almost nothing dying and sinking to the bottom of the ocean to feed the microbes in the ocean sediments. So ocean sediments barely accumulate. They accumulate very slowly, and there's just not a whole lot to eat. And so because of that, we can calculate, we still find, now I got to remember whether I actually put the slide in. I think I didn't, and I should have. But we can still measure decreases of oxygen. So out here, there's about 8 meters of sediment. So like 30-ish feet of sediment before you hit basement rock. And that 8 meters of sediment has been accumulating for about 50 million years. And so we can calculate from those kinds of rates that the lifetimes of these cells might be something like 10,000 years. A cell might live for 10,000 years before it reproduces. And the research question there is, WTF? How does that work? It is absolutely astonishing that that can happen, and we don't really know how. You know, again, my question is, what are these cells eating? And so one thing I do is measure the activity of digestive enzymes that these microbes produce to break down the complex organic matter that they need to eat food. And these are just exactly the same kind of enzymes that you might have, that you do have in your stomach right now. And for what it's worth, these are measurements not from the South Pacific Gyre, but from deep sediments in the Baltic Sea. So here we're going down to like 80 meters below the seafloor. And what you can see is that basically these organisms are eating the same stuff that you and I are. So closterpain and gingipain are these enzymes that break down complex proteins. And gingipain actually is named after gingivitis, because it is the same enzyme that the gingivitis bacterium uses to break down your gums if you don't brush your teeth. And you've got these in your stomach as well. So these microbes are eating proteins, they're eating carbohydrates, they're eating a little bit of fats, just like we are. But again, how do you stay alive? And you know, again, we don't know, but I want to bring you back to this figure from my colleague Jordan. who found a really interesting thing in these same sediments that these enzyme measurements are from in the Baltic Sea. So they reconstructed the metabolism of this type of organism called Atrobacteria. And what they found is that this organism can synthesize, even though it's living at very low energy, it can synthesize just about every different kind of amino acid, which is kind of amazing because it takes a lot of energy to synthesize an amino acid. And then it's got amino acid exporters. So it's got proteins that sit in its cell membrane and pump out amino acids. So it's barely, oh, and I should say, it also has it produces a lot of digestive enzymes. So it's bringing in complex food, taking the energy from that complex food and synthesizing amino acids more or less from scratch, and then apparently pumping those amino acids back out. Now, why would you do this if you are, living at the energy limits of life, right? So little energy that takes you 10 or 100 or 1,000 years to reproduce. Why would you feed the rest of the community? And the answer almost certainly is that when resources are scarce, the whole community helps by sharing. So what we think is that these organisms, these Atrobacteria, are in a symbiosis with other species. So they are probably feeding other species amino acids and getting something back. And we don't know what they're getting back, but we think it might be some kind of vitamin or some kind of what we call a cofactor for an enzyme. So something that helps your enzymes work properly, or something else. It could be anything really. But I kind of love this finding because it's, nature has been working for 3+ billion years to figure out how to keep life going when resources are scarce. And in this case, it looks like the ecosystem has fallen on the solution that you keep life going by sharing your resources. And that's I'm not often involved in papers that have such a clear moral to them, but it's hard to not see that in this paper. Okay, so that's a lot of the science that I'm going to talk about, and I want to talk a little bit about just sort of what the life is like going out and collecting these samples. So my PhD is in marine science, and I do research at sea when I can. The federal government operates a research fleet, and this is just a small number of the research vessels that they operate. It's the JOIDES resolution I talked about. These vessels range from the enormous, so this is the RV Sally Ride, which can stay deployed in the Pacific for months at a time. It's called a global class vessel, so it can sail anywhere in the world, and it only returns to its home port in San Diego maybe every few years. And they range down to the smaller. This is the RV Endeavor, which I'm going to talk to you a little bit about, which is, more stays out for weeks at a time. It can go out into the middle of the Atlantic, but mostly it'll stay within usually a few hundred miles of shore. And even though it's not huge as research vessels go, it can fit a fair number of scientists. I think this is a cruise, I think we had a dozen scientists aboard. And of course, we're all on the same ship, and we're all doing different research. And so how do we do that kind of research together? And the answer is we work together really, really closely. I got some advice, and I don't really know if it's true, but I think it might be. I got it from an oceanographer. An oceanographer said that oceanographers study similar things to geologists, but oceanographers tend to be more pleasant people. And the reason is that if you can't get along in a group, nobody will invite you on their ship. If you're a geologist, you can go out into the woods by yourself and go knock out rocks to your heart's content, no matter how big of a jerk you are. But oceanographers really have to be team oriented because ultimately we're all stuck on these ships together and we have to help each other succeed. So a lot of life on these research vessels is having a ton of meetings. But another piece of life is going out on the deck of the ship to collect samples. So we can collect samples. We collect a lot of different kinds of samples. we use this device here called a CTD rosette to collect water samples. So the way these things work is we deploy them off the side of the ship and send them down into the water and close them off at whatever depth we want, and then we can bring it up, and then we have a sample of water from that depth. This is also outfitted with all kinds of instruments to measure depth and salinity and temperature and all kinds of other parameters that we might be interested in. We can deploy coring devices to take cores of the sediment at the bottom of the ocean. And then we do a lot of experiments. So I tend to do a lot of incubations of seawater and sediments at sea. And so a lot of times what I'm doing at sea is making little measurements and writing down numbers for many hours at a time. And that can get exciting because, of course, on ships, there's weather, right? I have a vivid memory of taking this photo because this is just the beginning of the wave crashing over the side of the ship. And about a second after I took this, it hit me square on the chest and about knocked me over. I like this picture because it's not totally dramatic at the beginning, but like you can see there's like a 10-degree angle between the ship and the horizon right there. So a lot of that time I'm, we don't do a lot of deck operations in those kinds of conditions, but if I'm in the lab, I'm trying to do my experiments while I'm doing this, which is I don't know, either you're into that kind of thing or you're not, but I have a lot of fun with that. And of course, we just see a lot of beautiful stuff, right? So, the sunsets at sea are amazing. Dolphins love to play in the bow wakes of the boats. I don't really have anything more to say about that, except for to say that it's fantastic. I think I have used up, well, I've got a couple more minutes, eh? Is that right? All right, so I want to talk briefly about the other kinds of extremes that I'm interested in, which is microbes in the cold. And to do that, I've worked a lot in this archipelago called Svalbard, which you can see is about halfway between the northernmost part of Norway and the North Pole. If you've read his Dark Materials trilogy, there's armored polar bears up there, and there are in fact polar bears. They're not armored. There's really two towns in Longyearbyen. The rest of these towns that are marked here are more or less a lie. There's Longyearbyen and there's Ny-Ålesund. Longyearbyen is a real town. It's got a skate park. It's got houses. It's got an elementary school. You're not allowed to be born in Longyearbyen, so pregnant women are required to go to the mainland in about their 8th or so month of pregnancy. But there's a hospital. The economy used to be mostly mining, and so you can see this like kind of leftover mining equipment, and now it's mostly tourism. And then there's Ny-Ålesund, which is really the furthest north research town, or really the furthest north town in the world, and it's purely a research village. So you have to get permission to go there. You have to be a researcher, or you can briefly tour if you come on a ship. But and there's not much to it. This is basically an aerial view of the entire town. Everybody eats in the cafeteria over here. Everybody stays at various national research stations. So there's an Italian station, a German station, Norwegian, Chinese, Korean, etc. And it's quite lovely. We've used this mostly as a base to go on to sail on the and collect mud from the fjords. And the thing about fjords is, these are landforms that are carved out by glaciers. And the way they work is a glacier goes a certain ways and it's pressing dirt and earth up in front of it. And then it gets to its maximum extent and stops and pulls backwards. And that leaves a lip. And so these fjords can be extremely deep. There's a image of a different fjord, but I thought it showed it nicely. They can be 250 meters deep in the middle, but these sills at the end might be only a couple of meters deep. So you can't go in with a big boat. So we use this boat that is not a research vessel. but it's got a really shallow draft, so you can get into these shallow fjords. So we'll go out on this boat for like maybe a week at a time, and that is a really interesting experience because the boat is not a big place. We spend a lot of time collecting water samples, collecting sediment samples with these cores, and working out in the cold. We get really muddy because processing these mud samples on a boat that doesn't really have any kind of like research facilities or sort of infrastructure to it is turns out to be a messy job. I think in this picture I was awake for like 30 hours or something like that. The researchers get to know each other real well, right? So the entire cabin of or the entire galley, at least, and stateroom of the ship is about maybe 12 feet long and not quite tall enough for me to stand up in. So we cook each other dinner. There's no ship's cook. We hang out close on these benches. I haven't shown a picture of the cabins, but it's just one V berth with about 7 bunks and no shower. So if you're on there for a week with a bunch of other scientists, you're going to see them changing their clothes and nobody's taking a shower. So that gets a little fun. But actually I love it because you sort of, for the reason I said before, because really it's a bunch of scientists trying to do science together in really close quarters. And so when the people are good, then it's a lot of fun. And of course we see just some like unbelievably beautiful sights while we're doing it. It's got to be the best office in the world. These are some beluga whales that I was lucky enough to see, and reindeer walking through Neolus Arctic fox. I took this picture from the cafeteria in Neolus, and I was in the bread line while I took it. I think maybe the coffee line. I think this is from the coffee line. Anyway, this little guy was like just outside the window of our of the cafeteria. And the polar bears, they're real. So this was a lot of fun. This was a fin whale, which is almost as big as a blue whale, had died. And because the water is cold, the carcass gets preserved pretty well. So this fin whale, and this bone you're seeing back here is a rib of the whale, had died actually a year before. And it turns out that, well, so polar bears had been feeding on the marrow from these bones for years. So this was a mama polar bear teaching her cub how to sort of swim and get to the good bones. And I'll tell you, I'm going to end this with a story that I think is kind of funny. The captain of this research vessel farm, who he's this guy right here, and he's lived in Svalbard his entire life. He announced that we were going to stay in a different, we're going to spend the night in a different place than we normally spend the night. If he announces that, then you don't ask questions why, you just accept that we're going to do it. And of course, the reason was that he knew that this fin whale skeleton was there and that we might see polar bears. And so I said, Stieg, after we watched these polar bears play for a couple hours, I said, Stieg, how did you know that this fin whale carcass was here? And Stieg said, well, I put it there. I said, Stieg, what do you mean by that? So Stieg had been doing logistics for one of the BBC Blue Planet, or rather Frozen Planet series. And so he'd been shuttling these camera people around. And he got a call that there was a fin whale floating dead in one of the fjords. So he told the Frozen Planet people about that. And they said, yeah, we'd love to take some pictures of that. And they got to it and they said, well, this is great, but it's not in a scenic enough of a fjord. And he said, well, do we need to move it? And they said, yes. And Stieg, being a Svalbard guy, says, I know how to do that. So he rigged a harness to the dead fin whale and towed it about 20 miles and then grounded it at this spot, which was judged to be sufficiently scenic by the BBC people. And it stayed there for about a year, and these, provided food for the polar bears. I don't really think I have a conclusion to that, except to say that this search for understanding what it is that allows microbes to live under really weird conditions in the environment has been a lot of fun for me so far intellectually, because there's a lot of weird conclusions to make, and I get to go to places like this. So do I have any questions about that?
Well, I would like to say thank you very much for taking time to speak with us today. It was very interesting. I enjoyed learning some interesting aspects of your research. I'll ask you, have you had any sort of dangerous encounters? I mean, you've been close to polar bears, you're out at sea. I mean, I think I've seen at least commercials for Deadliest Catch that makes it look really dangerous. Have you had any dangerous happenings?
You know, a thing that I like, particularly about the UNOLS fleet, so this is the Federal Research Fleet, is that these captains are used to working with scientists. And on these ships, the scientists, the chief scientist's word goes unless the captain vetoes it. And so not long after that wave picture that I took, the captain declared that deck operations were not going to happen. And, you know, the thing I love about that is he doesn't really care about the science going on, right? But he does care about the safety. And so I think the community has a pretty big, a pretty good sense of safety for this kind of things. And occasionally you'll hear about people who don't make great safety choices, but I think I've generally been able to stay away from safety problems by being conservative about them.
Well, very, very good. I ask maybe a more technical question now.
Sure.
You use molecular tools to look and get an idea of how they are functioning in the environment. Is there any room for interpretation there? I mean, just because you detect a gene, does that necessarily mean it's operational in that environment?
Nope, sure does not. So we use a lot of other tools. I didn't get into it, but we try to use looking for presence of DNA, looking for the presence of the corresponding mRNA, so messenger RNA, looking for the corresponding proteins. And then I showed you some of these enzyme measurements I made, which, you know, theoretically show that the enzyme is actually active in the environment. You know, to me, none of those methods are perfect, right? All of those methods are kind of weak, actually. So the thing to do is to bring together as many different lines of evidence, you know, including what we know out of cultured species and so forth, to tell a coherent story. I think you can't really learn anything from just one line of evidence. You can maybe make some hypotheses.
I have one question.
Yeah.
I've heard on occasion of climate change and warming up north causing potential problems either in disrupting ongoing research in some way, or I think it was in Russia where they said that defrosting permafrost was thawing out potentially dangerous pathogens. Has that impacted your research, or do you see that impacting the field?
I was so mad about that report about the pathogens because it's nonsense. It is true that thawing permafrost, that you can get live organisms out of permafrost that has been that has been frozen for, you know, tens of thousands of years. But the thing is, there's not a ton of pathogens just sitting around in sediments, right? So that's not a concern. But the permafrost thaw is an enormous concern. And in fact, I mean, I think it's kind of telling. A big chunk of the cafeteria, so a bunch of my colleagues were up there just a couple of weeks ago, and the cafeteria building was partly shut down because the permafrost it's built on was failing. So, you know, we're up there trying to investigate permafrost thaw, and you couldn't eat the way we normally did because the permafrost thaw. So you're right that logistically it is a big problem. But, you know, it's a problem for the carbon cycle too, right? It's releasing a lot of this organic carbon and potentially methane that had been sequestered, and now it's going out into the oceans. It's, you know, sloughing off into rivers, going out into the oceans, and becoming available for microbial activity. And that's going to enter the carbon cycle and add to carbon dioxide in the atmosphere.
Thanks.
Have there been many opportunities for you to involve students in some of these trips and to faraway areas?
Yeah, so my graduate student, Brianna, went to, I shouldn't say my graduate student, but a graduate student working with me, Brianna, was in Svalbard, like I said, for most of the month of March. And I started going there as a student. And yeah, so we've had students that go. One neat thing about the IODP expeditions, these 3-month expeditions, is that you can just apply to sail. So if you are a scientist, you don't have the project, you can just apply. And if you have a skill set that they need, you know, they try to get together really multidisciplinary teams. If you have the skill set that they need, then you may get accepted to go. So definitely graduate students from UT have sailed on IODP expeditions without that nobody at UT was previously involved in. So it's a big priority to get students to the field.
Okay, great. I bet it's an amazing experience. Do we have any other questions out there? All right, not hearing any. I want to thank you again, Dr. Steen, for sharing with us some of your information and adventures. And I do want to thank you. We very much enjoyed it. I also want to make sure that we'll give a big thank you to John and Lainey, who make this possible, arranging all of the logistics and making sure that everything is smooth sailing, so to speak. And then I do want to put in a plug, ask everyone to join us back next week, invite a friend, visit LMU or the School of Math and Science on Facebook or Instagram, and make sure you register and come back next Wednesday, same time, where we're going to listen to Dr. Stephanie Koster from Randolph-Macon College, Close Encounters, Field Adventures of a Wildlife Biologist. So with that, I thank everyone for attending. Go have some fun, go have some adventures, and we'll see you back next week. Thank you.